morning, students.
Good morning, sir.
How are you?
Fine, sir.
Okay. Second year, sir. First year, sir.
Second year.
Right. So, all syllabus complete ಆಯ್ತಾ?
Yes, sir.
Any pending?
No, sir.
Right. So, before starting the lesson, first of all, here are the development of embryo. If you observe this one, can you guess what is the name of the lesson? Right. So before discussing the development of dicot embryo, we have to see what is the. So first I will say my self introduction. So myself Krishnamurthy, I came from Telangana. Right now I am working in Chitradurga district. Okay. So today just I will explain you what is the development of dicot embryo. It is a very small and it will be very interesting also. So before discussing the development of dicot embryo, once observe what is the main important characteristic feature of the angiosperms. Enadu flowering plants characteristic unique feature. First characteristic feature is enadu that is common. First characteristic feature of the angiosperms flowering plants. Second one no double fertilization, right? So what is a double fertilization? Right. Actually, our topic is development of dicot embryo. But before discussing the development of dicot embryo, we have to see what is a double fertilization. How the fertilization is occur. So unique character of the angiosperm is double fertilization. So what is double fertilization? Double fertilization is equal to syngamy
Plus sexual fusion.
plus triple fusion. So fertilization occur here twice. So hence it is called double fertilization. What is syngamy and what is triple fusion? Once you have to observe. So because of when we are going to discuss in the development of dicot embryo, sometime we will discuss the terms of endosperm. So how the endosperm is formed we have to see. So the endosperm and then where is the polar nuclei? So that we have to discuss in later itself. So first of all, what is syngamy? Syngamy ಅಂದ್ರೆ ಏನದು? Right. So what is this one? This one. Ovary. Next this one. Style. This one. Stigma. Right. So whether it is, for example, the right type of pollen rings forms on the stigma, then pollen will be, so means stigma will be accepted. Then pollen will be germinated onto the stigma, which is comes to the tissues and then which is comes through the style and reach to the ovary. Next, in the ovary, it is ovule, right? The pollen tube is enter into any one of the way, like porogamy is there, mesogamy is there, chalabogamy is there. So whatever it is, the pollen tube will be reach the ovule and enters into any of the way. Then what is the part of this one? It is the antipodal cells. Then these two secondary polar nuclei. Next down there is the three cells are there. Middle is these two are synergies. Okay, now this pollen tube which is carrying how many male gametes? Two male gametes, right? So these two male gametes which is carry like this, and these two male gametes which is enters into the ovule. So next, these male gametes enters into the ovule, then immediately what will happens? So due to the endosmosis, two male gametes will be means ruptured. So due to the rupture of the pollen tube, then immediately two male gametes are released. So now one male gamete fused with secondary polar nuclei. Okay. Next, one male gamete fused with egg. Okay. So one male gamete fused with secondary polar nuclei is called what? Triple fission. And one male gamete fused with egg, it is called syngamy. Collectively these two are called double fertilization. Fertilization occurs twice, hence it is called double fertilization. So what is the largest cell in this? Largest means biggest cell in this. I actually forget one label here. Cell. Right, central cell. Very good. So this is a central cell. Okay. So this is during the fertilization. After the fertilization, what will happen? So these antipodals will be degenerated or not degenerated? Right. So now, for example, here the 3 antipodals are there. Now 3 antipodals are degenerated. Right? Next here, out of 3, 2 will be. So 2 synergids will be degenerated. Right? What will remain? X. Means it is already fertilization is occurring. It means what? Zygote. Next. Here is a 2 polar nuclei. Okay, 1 polar nuclei. So 1 polar nuclei becomes to what? So after fertilization becomes to PEM. Full form? Right. So then central cell becomes to what? PECL. Endosperm cell. Right. Primary endosperm nucleus and primary endosperm cell. So both are what? Endosperms. What is the function of endosperms? Provide the nutrition to the developing of embryo. Right. So zygote. So in the so this is ovule, right? So now in the ovule there is no any other cells except only zygote and then endosperm. Only zygote is there and endosperm. That's it. Okay. So everything will be degenerated. At the mature embryo cell, how many cells are there? How many nuclei are there?
7
Right, 7 celled and 8 nuclei. Right. Sometimes they will ask in PUC also, why the so mature embryo sac is 7 celled 8 nuclei. So you have to be right, be careful. Right. So this is the double fertilization. Next, after the double fertilization, what will happen here? Dicot. So and one more remember, we have 2 embryos are there, dicot embryo and monocot embryo. What is the dicot and monocot? Monocot andre enadu? Having only one cotyledon. Dicot andre? 2 cotyledons. Right. So we are discussing the dicot embryo, means development of dicot embryo. Okay, monocot embryo, it is not given in NCERT, no need to worry about it. We have to focus on only majorly dicot embryo cells, right? Next, so this development of dicot embryo is discovered by Hansteen. So scientist name is Hansteen in dicot plant of Capsella. Another dicot plant of Capsella, right? So already I said the zygote is formed. Will the zygote will be divided immediately? After the fertilization, will the zygote will be divided immediately? Why? It requires what? Endosperm. Okay, without endosperm, the zygote will not divide. Okay, right. Now here observe. So this is the zygote. Diploid or haploid? Diploid. Right. This is zygote. Now I will explain it here. So this is zygote. I am taking only one zygote, not the remaining parts like embryo sac and other primary endosperm cells. Right. This is a zygote. Okay. Next. So zygote will be divided. If it is gets little amount of endosperm is formed. If little amount of endosperm is formed, immediately zygote will be divided into next embryos. Right. Now here is a zygote. Next, this zygote will be becomes to what? If it is getting nutrition from the surroundings, like PEC and PEM from primary endosperm nucleus. Means from endosperms, if it is get nutrition, then it will be take the nutrition and becomes to increase in size. So first zygote, once see the size of the zygote. Now you have to see the size of the zygote. Okay, this is after fertilization the size of the zygote. Then whenever it gets the nutrition from the endosperm, then immediately it is increasing in size. So then the size of the zygote will be increased. So now the zygote is called oospore. In NEET also they will ask here sometime how many mitosis and how many meiosis occur in development of dicot embryo. we know simply there is only diagrams we know, and next in PUC bug you know. So whenever they ask the question, then we will write the answer with the diagram still. But how many mitosis are there, how many meiosis are there, we don't know. Right. So what is the mitosis? Mitosis from the mitosis how many cells? Right. From the mitosis how many cells we can expect? 2 cells or 4 cells are? From mitosis how many? 2 daughter cells. Okay, 2 daughter cells. Then from meiosis, right, it is already in there in first year. What is the lesson name? Right, cell cycle and cell division. From mitosis there is a 2 daughter cells are formed. From meiosis 4 daughter cells are formed. So meiosis occur in our body which cells? Gametes. In female and males also the gametes only, sperm and ovum only, which can able to differentiate or form by the meiosis.